So far, I really as much as that I know I really can't tell her no stuff. I'm afraid to ask my name. But don't. I'm not even like, I'm
Also, how could I miss my favorite chapter, y'all?
I thought you'd say favorite class.
Favorite class too, definitely, yes. Yes.
We don't do Kahoot today?
The what?
No Kahoot today?
Yeah, we're gonna do it. I'm just getting everything set up.
Okay.
Okay. So I'm gonna get this Kahoot set up, and because I can honestly use any ounce of energy that I can keep. I'm probably just gonna put it on a game today. Now chapters, because it's over chapters 9 and 25, and we know what we need to know for that, because I only went over what we needed to know. Believe me, I covered everything you needed to know. And so there might be questions on this. I'm just gonna warn you guys that when I had written them, I had intended to go over everything in those chapters. So there might be some stuff on there that if we didn't go over it in class, it might still be in the questions because I didn't edit them, but you don't need to know it if we didn't go over it. Okay. So really, I was honest about that stuff. That's what you needed to know. I'm going to put it on as a game just because I need to not pass out when I'm giving the lecture. I need as much energy. Yeah. It's like really tired is how I feel. And like hot.
Are you taking antibiotics?
Yeah.
What kind of question is that? Your microbiology teacher.
Yes.
Antibiotics?
Yeah, I'm on antibiotics. I'm about halfway through. Okay, so I don't even know what I'm doing right now. There's 9 and 25. I'll just do 9. Okay. Let's do the chill one. I know everybody likes that one. I usually like 5 minutes. Let's do 5. Yes.
Are multi-select Kahoot questions premium only? Because my group is doing a Kahoot quiz.
Probably. I have
Two of my questions are multi-select.
I have a pass that I can give out to students if they want to make their own, but I don't know how it works. But I can try to give it to you guys and you can see if you can figure it out and make it work. Email me and that way I won't forget about it. So otherwise I just like I'll hear you and then like you'll be like, hey, about the thing. I'm like, I know you said the thing, but and then I will forget about it.
I think it's the I don't know names. It's the one with the pink hair that has the Kahoot account.
Okay. Yeah, I mean, I'll just yeah, you guys, whoever emails me and then you can just send it to her.
I'll ask her to email you about the pass.
That sounds fine. Yeah. That might be what you guys need. If not, then I don't know, we'll have to find a workaround or something, but yeah.
I'll let her know right now.
I was going through all this stuff yesterday.
Right. Yeah. So yeah, which if you can figure it out, but like at the same time, it's one of those things where it's like it'll probably be figured out by deduction anyways, like when you submit the other ones. I look and like I'll look over them. When you guys submit them, like I'll look at yours and like when your paper or look at your paper and like if what you're putting on there makes sense to me, then I just give credit. But like I don't always look that it matches the thing. I really wouldn't worry about it as long as it gets submitted. Yeah. Yeah. Yeah, at least look and make sure they make sense. That's pretty much all I do with those though.
I know every day. With all this fresh on your brain. I will bring it if you vote. That was like, literally, I'm sitting in class and spending my no. Don't quote me. I like Breno.
All right, everybody in, it's gonna be in.
I'm not in yet.
A little bit, couple more minutes. All right, we good? Remember, you guys can always join in if you need to, especially on these chill ones, like it's pretty easy to just pick up. So you guys know how this one goes. I'm gonna let you guys have at it. Oh, yeah. Let's. It looks like this one here has some sort of arches. It reminds me of Egypt. Looks, I don't know what Egypt also to me for some reason. Anyways, have any preferences on these ones we haven't done yet? This one? questions about any of the stuff that was on there? Was there any weird stuff? Because I didn't look at it. Was okay.
No, there's something weird.
Okay.
Everything was from chapter 9.
Yeah, I did chapter 9 because it had the most. I had 2 separate ones. I figured I would do chapter 9 because that's the most stuff. All right. So good work, guys. So we'll get into chapter 10. Get away. So finally, it's chapter 10. This is my favorite chapter, which might surprise people since I really like infectious diseases, and this is definitely not that, but I feel like the stuff in here is just really cool how it explains things. And so if you guys are in physiology or have taken physiology, you're probably either going over this stuff or already went over this stuff, and so you might have an idea of how this works. Hopefully this helps solidify that. This is your first time hearing this stuff. I hope it is exciting to you as it is like it was to me when I took biochem back in the day. So, okay. All right. So we're going to be discussing in this chapter the metabolism of microbes, but honestly, a lot of the metabolism but pathways apply to all organisms. I always close it right before somebody walks in. Sorry. All right, so metabolism just refers to all the chemical reactions and physical workings of the cell. You could also think of it as when we're talking about macroorganisms, multicellular organisms like ourselves, all chemical reactions and the physical workings within the organism. So anabolism, there's 2 parts of metabolism, right? Anabolism and catabolism. I'm gonna tell you how I remember these, and they're stupid. Well, maybe not always stupid, but I don't know. Catabolism was kind of stupid, okay? So catabolism is breaking the bonds of larger molecules, and we should know, probably having gone through chemistry by now, when you break bonds, you release energy, right? Because you need energy to make bonds, right? Just like building things. If you're building something, you definitely have to put in a lot of energy, but it's pretty easy to kick that Jenga tower over. It doesn't take a whole lot of energy to do that, right? So catabolism is breaking the bonds of the larger molecules to release energy. And the way that I remember catabolism is the word for it is I think of cats, and cats tear things apart. If you have a couch and a cat, you probably know what I'm talking about. So cats can, I say, they're breaking things apart, ripping things apart, destroying things. Catabolism is breaking things apart, destroying the bonds, destroying these molecules, all right? A lot of us have probably heard of anabolic steroids. Anabolic steroids are molecules that you can take to help encourage your body to help build muscle, for example. So that's building, that's creating. We're building bonds, we're creating new things. Anabolism is biosynthesis. This is going to require energy input. It's building. With or without, I'm just saying, with or without the addition of steroids, anabolism, no matter how you're building, anabolism is building. So metabolism in general, we're going to be looking at the pairing of those 2 things together. Obviously, you can break down some things into the parts and then use those parts to build other things. You can use things that were built by something else to break it down to get the energy out of it. And all of these different things we can use to create ATP, which is our energy source. The whole point of this chapter pretty much is getting into talking about how we make our ATP, the most important energy source of the cell. Remember, thinking back to chemistry. So first we're going to get into enzymes and kind of what they are and how they work. Getting into chemistry, we talked about catalysts. And a catalyst is anything that helps a chemical reaction go, overcome that, what they called activation energy, right? The energy that you needed to overcome, to get over that hump, to make the reaction happen. A catalyst helps with that, helps overcome that energy. And an enzyme is basically a biological version of that. So an enzyme is a biological catalyst. So it's going to be an organic molecule used typically by cells that is going to help them overcome these activation energies of whatever reaction they're trying to get to, right? So they help increase thermal energy, which is going to be increasing basically heat, literally the temperature. Because remember, if you increase temperature, heat is like the most basic form of energy that we have, but it does work. It excites, molecules and helps energies to go. So it helps with that. And we can also increase the concentration of reactants to help things go as well. And then we can also add in other catalysts, if we have multiple catalysts and a whole big old line of reactions. So we kind of already know that because we've talked a little bit about how that might work for bacteria, like in operons and crap like that. So we know they can tie a bunch of things together to help it go. All right, so enzymes. How are they themselves going to be getting that job done of overcoming that activation energy, being a catalyst? They will participate in the changes that are going on to the molecule they act on, but they do not themselves change permanently. So when you do use an enzyme in a reaction, the enzyme will not be used up. That's a good thing, right? So your cell can keep going over and over again using that enzyme. So it should not be used up permanently, even though it can help in changing its molecule to its product. The thing that it works on, the molecule that it acts on, is called a substrate. So an enzyme binds to a substrate, and that's what it's going to act on and create whatever the products are in the end. All righty. Most enzymes that we are going to learn about, and probably the only ones I'm ever going to talk to you about, are going to be protein molecules. You can have some simple enzymes that consist of just the protein by itself, just like only protein, and that's it, and it functions, and that's all it needs. However, I'm not gonna lie to you, most of them are conjugated enzymes. Conjugated enzymes mean that they need some other thing to help them do their job. You guys know about hemoglobin, right? So that's a protein molecule in your red blood cells, and it interacts with, iron. It has iron in it. And if it didn't have iron in it, it can't do its job, right? It can't interact with oxygen the way that it needs to transport oxygen around to your body. So we have iron in there as something that's essential for hemoglobin to do its job, right? It's the same concept with these enzymes. So think of them as being like the hemoglobin, and that the iron is the part that they need to work, okay? And then the oxygen would be the substrate, okay? So we'll get into it. We'll go over some of these and talk about what this all means. But okay, so if we have conjugated enzymes, so easy enough to imagine that if you have an enzyme that's just a protein and it works just fine on its own, great, you're an enzyme, cool. But when you're conjugated enzymes, you have parts. Now we've already established that if we had hemoglobin, that would be the protein part. And then the iron that is bound with it, that would be the thing that's bound with it. And in this case, it would equate to the cofactor. Okay, so the whole functioning enzyme, if it were an enzyme, we would call it the holoenzyme. Whole enzyme, whole functioning enzyme, holoenzyme. Yes, you can equate that by how it sounds. I feel like that's the best way to remember it. Then that is broken up into two parts: the apoenzyme, which is just the protein portion of that holoenzyme, and that cofactor. Like I said, if we were dealing with hemoglobin, we would be talking about hemoglobin, the protein itself being the apoenzyme and the iron being the cofactor, right? And then our oxygen that comes in, and that what's going to be doing the work, the thing that is affected by all of this, is going to be our substrate. That's an example of how, proteins would be working. Now, so you can have enzymes bind with other organic molecules or with things like metal inorganic molecules, like metal ions, like iron, right? So iron falls into that category. It's just a regular plain old cofactor. But if you are an organic molecule, like a lot of our vitamins are, then that's a coenzyme. So anytime it's organic molecule, it's a coenzyme. If it's a cofactor that is organic, coenzyme. That usually is going to include a lot of our vitamins, like, B12 and stuff like that. They're organic molecules. They're not proteins, but they're organically based molecules. And they usually, the reason we need them is because they're going to bind with our proteins to help them do their jobs in this way. Okay, so that's how that's all going to work. This is biochem light, y'all. Okay, so the apoenzyme is where the actual action is going to be happening. It's just that cofactor is going to be what's helping it do its job. It needs to be there for it to function. So now that we know the apoenzyme portion of the holoenzyme, the whole functioning part, is the part where our substrate is going to bind. It's the protein portion with the three-dimensional structure that's important for how it works. And every single enzyme compared to one another is going to have a primary structure that's different, how it folds different and its unique active site. So the reason we have the different proteins, basically, sorry, is simply because, what did I click on? Whatever, I don't know. The reason we have the different proteins is because they're all different. They all are, shaped different. They all function different. They all do these different things. Okay, so that's why we have to have a whole bunch of them. Why DNA polymerase? ACE, anytime you hear ACE, that's an enzyme, okay? DNA polymerase is different from RNA polymerase, right? Because they have to interact with different nucleotides. They're making different products. So even though they have similar functions, they're going to have different structures, different amino acid sequences, how they're folding is different, everything like that. So that's all this is talking about. Those three-dimensional structures of the enzymes can be almost more important than the actual chemical makeup of these enzymes, simply because if I were to replace an amino acid in a protein sequence and it still had roughly the same properties and the structure is still shaped the way that it still is shaped, it can still interact the same way that it would interact. Who cares? Like, that's still working, right? So that's the idea. So sometimes the exact molecular makeup may not be as important in this. This is not like chemistry in that regard, right? When you guys learn about this stuff in sort of inorganic chemistry concepts, it is like exact chemicals leading to exact chemical products. These guys are like bigger picture stuff. These are interacting with not just the chemical forces that are going on here, the actual structural interactions and how that's based on the overall structure of the protein versus the substrate and everything. So it's big picture stuff. We're talking about almost looking at it as like, how these things are going to fit like a lock and a key, like it shows in the picture. So the lock and key thing might be more important than the exact chemical thing. Right, we already kind of introduced this concept of metallic cofactors, but we know this could be things like iron, copper, magnesium, manganese, zinc, cobalt, selenium, and all sorts of stuff like this. These are why we need these metals in our diet to some degree, right? We know we have to have them in our diet. We know that they're present in our diet. We don't really focus too much on them because they're usually kind of just there in our diet, but we need them. And this is why. It's because it helps with a lot of our enzymes to help them function. Oftentimes they can themselves participate in the reaction, or they can just help with maintaining the structure of the enzyme portion itself. So the protein structure can be helped into its correct formation by having that metal ion present. That can be true of the coenzymes as well, but they're just going to be organic versions. And they can often work with the actual protein at the same time. So almost as like a partner in the process, more so than the metal ions are just kind of there to help support like the structure a little bit and maybe provide a little assistance with their chemistry. But the coenzymes, them being a little bit larger, being organic, they can actually work along with the process. So vitamins, like I said earlier, are usually an important part of coenzymes. So there's 6 classes of enzymes. Some of these might not be any surprise to you guys. Honestly, if we break these down and we learn about chemistry and we learn about biology, these aren't really any big surprise. They just have nice fancy names. The first class is going to be the oxidoreductases. And tell me that y'all don't know what a redox reaction is. We're going to be getting into that today. So I hope you're excited. Okay, so oxidoreductases. Oxidoreductase, or if you guys are thinking of redox reactions, so reduction and oxidation, right? All of these things. Lump them into one area for me, please. If you hear oxidation, if you hear reduction, if you hear redox, if you hear oxidoreductase, all of them involve exchange of electrons. That's the biggest thing to connect to those guys, okay? So if you ever hear anything related to those terms, it's always going to involve exchanging electrons, always, right? You guys remember hearing about these, about losing electrons and gaining electrons and all those things. So anything dealing with that falls into that category. Next, we have the transferases. Kind of tell you what they do, right? ACE means enzyme. Transfer, what are we going to do? Move a group from one area to another. That's all. Transferring. Next, we have the hydrolases. These guys cleave bonds on molecules, but we're going to add in water. That basically means we'll take water and like use it to like seal up the ends that we would be opening up. So if you're breaking a bond between two things, you can't just leave like nothing on there. You need to add on molecules to make up for what you just broke, right? So we can involve water into that reaction, and it's sort of like the H at one side or the OH at the other side help contain like this what you just split apart. It's pretty common what you would see in breaking down large polysaccharides like starch. And we'll learn about this when we go into the lab, when we look and see if our organisms can break down things like starch. And if they can, that means they are able to, they have a hydrolase called amylase that can break down starch into its components, which are sugars, just regular sugars. But when you break them apart, got to do something about that. They don't just break and that's nothing. So hydrolases do that by adding in water. Liases are things that are going to add groups or remove groups from double bond substrates. Never going to ask you about it, by the way. Isomerases, because I feel like it gets too complicated in the chemistry of it, and I don't want to focus on that. Isomerases change a substrate to its isomeric form. So if you guys remember about isomers, it's just like different versions of the same larger molecule, right? It can just be like mirror images or something like that. They can even have different functions in the body, but this is just an enzyme that helps switch between those. So if you like think like D or L oriented amino acids, so if you ever heard of like, you know, D cysteine or something like that, or L cysteine or L alanine or something like that, those are just isomers, the D or the L orientation of them. These guys can switch them. That's what isomerases do. All right, ligases. These guys catalyze the formation of bonds. You can think of hydrolases. We're breaking bonds, adding in water to kind of help that process go. Ligases are creating bonds. By obviously, if you're creating bonds, you have to put in energy, right? So that's ATP. And then removing water. So think of it as if we had taken those sugars that we'd split apart and we sealed it up with our little water molecule by splitting the water. Now, if you want to put them back together, what are you going to have as an output? Water, right? Because you put it in. So this is literally the reverses of one another. They're just reverse ways of looking at them. It's easy to break a bond. It's not easy to create a bond. So creating, like we said, building things takes a lot of energy. So you're going to need ATP to build. That's why ligases have the ATP. So you can think even of, if you wanted to go back to our terms that we learned earlier, catabolism and anabolism. Catabolism would be going on with the hydrolases and anabolism with the ligases. So we start fitting those concepts into those categories. All right. Yay. Okay. Oxidation and reduction. I don't know if this has the little thing where we talk about like Leo says GR or oil rig or whatever it is you like to remember these things by, but I learned it as Leo says GR. I'm going to tell you what that means. If you have no clue what I'm talking about, I'm going to tell you what that means. This is getting into oxidation and reduction, which, like I said, has everything to do with electron exchange. So these have to do with one molecule has to lose an electron in order for another molecule to gain an electron. That's just, you know, science. That's physics. That's the rule. Matter cannot be created or destroyed. So you have to have exchange going on. So if you want an electron added into here, it had to come from something. That thing had to donate it. Right, so this is how I remember it. Leo, the lion, let's say, Leo says GR. So I say this is lose electron. I learned this literally. I learned this in AP Chem and I've never forgotten it and I can't forget it. Now it's like with me forever. Lose electrons means you are oxidized. Gaining electrons, I'm just put E minus, because you know what that means for sure, means reduced. So that's one way, one little way you can remember which one's which, okay? I have another one for you, which you might like better, which is oil rig. I have to think through it sometimes because it's not the way I learned it, so be patient with me. So oil rig, at least that's like some concept we can get on board with, right? Obviously, the O is oxidize. Oxidize is loss. Reduction or reduce is gain. So whichever one works better for you. These are just, and if you have other ones that you learned that work better for you, fine, that's good too. But here are a couple of options that if I ask you on the test, if you're oxidized, what does that mean? Then you would be able to write down oil rig or Leo GR and say, oh, if we're oxidized, that means we lost an electron. Okay. a lot of these concepts as we're going forward, I'm going to be calling things as like, okay, this reduced this molecule, blah, 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 blah. And it's easier to keep up if you kind of can keep up with what does what. So oxidation, we're talking about loss of electrons. A compound that loses electrons, we call it oxidized, okay? Reduction, we are gaining electrons, and a compound that has gained the electrons is called reduced. I'm more concerned with you guys being familiar with these concepts of oxidation, meaning loss of electrons, and reduction, meaning the gain of electrons, so that whenever I'm not going to give you a big old like equation and be like, which one lost electrons? Now, which one was actually oxidized? Which one was oxidized and which one is, whatever. I'm not concerned with that. I want you guys to get the concept down more than anything, okay? So we can keep up with this. We're about to get into some serious biochem. And we're going to talk about some coenzyme carriers. You guys remember what a coenzyme is? The coenzyme, again, that organic molecule that helps the enzyme do its work. So these are coenzyme carriers. They are going to carry electrons for us, which is extremely important in creating ATP. It is the whole driving concept of this whole situation. This is so important to your body and for bacteria, of course. We love bacteria too. So anyways, we're going to get into that. Those guys are some molecules that can carry electrons for us. All right, so let's also talk about some other kinds of enzymes, exoenzymes versus endoenzymes. Exo always means outside and endo means in, right? So exoenzymes, just enzymes outside of the cell that do stuff outside of the cell, like breaking things down smaller so we can bring them in to the cell. Like we talked about with saprobes, right? They have to break it down to bring it in. Then we have the endoenzymes. These are enzymes that are contained inside the cell and stay inside the cell and do their work inside of the cell. Anytime we're talking about metabolic pathways, that's going to be inside of the cells. Okay. We have constitutive enzymes that are just kind of always around, always doing something because we always need them to be there. And then we have regulated enzymes. This, no matter what the type of organism you're talking about, we can regulate the production of any enzymes whatsoever. But this does come back nice and full circle for us to those concepts of those operons. So for bacteria, they have operons and they can induce or repress multiple genes at once. And that's why it's called an operon. But we can do that with our genes. We just have to do them one by one. We can regulate genes or we can have them constitutive. If we are constitutive, it doesn't matter what's going on in the cell. You're always making the same amount of enzyme. It don't care. If they are regulated, it's going to respond to need, right? Supply and demand. So if we have just a little bit of yellow going on, we have enough of these purple guys to break them down. If we have a whole lot, we're going to make more purple guy. Or if we have a whole bunch of purple guy and we've broken it down and now we don't have as much yellow, we don't need as much purple guy anymore. So that's really all this is talking about. It's just responding to what's going on in the environment, making more or less enzyme based on that. And this is going through that process of repression or induction. It's just a matter of if you're dealing with one gene versus a group of genes. And if it's a group of genes, that's only ever going to be in bacteria, and that's called an operon. Okay. We don't have operons. Okay. Oh, okay, so when we're talking about these enzymes and what they can do in disease, a lot of times bacteria can make enzymes that kind of allow them to infect us more easily, right? And we would call those virulence factors. Anything that helps a pathogen infect us or create disease, that's a virulence factor. So these things that increase pathogenicity, which is the same thing as what I was talking about, increasing disease. These could be things like streptokinase, streptolysin, elastase, collagenase, lipase, and penicillinase, which might not sound like anything useful to you when you hear it at first. But if I tell you, like, looking at the words, penicillinase is an enzyme that, what do you think it does? Breaks down penicillin. If a bacteria has penicillinase, that's a problem for you if you're going to treat it with a penicillin-based antibiotic, right? So that's what we're talking about here. Like collagenase, we know we have collagen in our bodies. Collagen is part of your extracellular matrix. It helps keep your tissues together and creates a barrier basically between the different layers in your skin as well. So if we can break down collagen, we can better get in between and deeper into tissues, stuff like that. So it becomes dangerous when you start looking at it that way. All right, we talked about denaturation with DNA. Same thing can happen with protein, right? If we heat something up enough, it's going to dissociate those weak hydrogen bonds and damage the overall structure of the protein. Same thing can happen with pH. If we have a whole bunch of hydrogen ions introduced or less hydrogen ions than what there should be in the area, that can affect how that protein's shape is able to function. So that can inhibit the substrate from getting to the site that it needs to, that active site. And then we can block the whole pathway that that enzyme is involved in, and that can lead to cell death, depending on the enzyme, right? Because denaturation, the different kinds of pathways in metabolism that we're going to be that you could look at, and we're going to be looking at some of them today. Linear, which is pretty straightforward. A goes to B to C to D to E as these steps are going on. This chemical reaction causes this product, then that goes through that chemical reaction, and so on and so on. The cyclic or cyclic versions, like the Krebs cycle that we're going to be talking about, you put something into this and it combines with molecules that are already there and then goes through all these molecular changes. And as it's going through, we lead to a product that will come and bind in with the next U, for example, and go cycle again and again and again and over again. So the products are kind of being recycled back in to bind with the next coming molecule. So that's how cyclic ones work. Branched, obviously, that makes sense as well. Like if you have a chemical reaction that leads to two products, then they can go down different pathways. That makes sense. Obviously, if you brought two things together to make one molecule, that's what the conversion ones would do. So that, right. So let's talk about inhibiting these enzymes. These inhibitors can interact at like places anywhere on the protein, but if they're interacting at the active site, which is the site where we normally would interact with our substrate. So if you're hemoglobin and you have a site and yeah, you have, say you have your nice little iron and that's working, everything's working there. Your oxygen comes in and can come and interact with you there, right? Wherever it binds with the iron or wherever, it doesn't matter, but wherever that site that it binds at is called the active site, right? So that's what we're talking about with substrate binding. If we have an inhibitor like cyanide come in and bind in that spot instead of oxygen, oxygen can no longer bind and it has is now unable to do its job, right? So we're blocking it. So that's competitive inhibition. Cyanide can come in and bind at the site that oxygen would. Cyanide has a greater affinity for that binding site than oxygen does. So it's very difficult to treat if you start having cyanosis develop from actual cyanide poisoning and dealing with that. So anyway, so yeah, you get it, right? So it's something that can mimic that area and bind in that same area and block what's supposed to be there from being there. That's competitive, right? It's competing for that same active site. Then we have non-competitive. So this one's a little hard for me to explain, but it's usually going to bind at a regulatory or allosteric site. So not the active site. Just think of it that way. Not the active site. Any other site that's not called active site, that's what I'm talking about, okay? So think of it as like you are an enzyme and you're happy and you're working and you like to carry, I don't know, markers from one side of the board to the other side of the board, right? You're happy to do this and you're happy to do that and whatever. Then suddenly something comes along that is your non-competitive inhibitor and binds to not where you handle the markers, but let's say a different part of the molecule. Let's say, you know, the non-competitive inhibitor in that case is like handcuffs or something. Handcuff me behind my back, and now I can't handle these well. It's not actually interacting with the site, right? It's just causing the shape change, so now we can't do our job the way that we were doing. Or like you normally carry, you know, basketballs from one end of the room to the other, and then something comes along that's really heavy on your back, and you now can't hold, just can't, like your back hurts and you can't carry anything anymore. Same kind of thing. So these guys will bind somewhere else on the protein, cause it to change shape, even subtly. But if it changes the shape and it can no longer bind with the normal molecule or interact with it the way that it's supposed to, that's non-competitive. So the difference between these two, looking back at this, right? Competitive versus non-competitive inhibition. Competitive, binding at the actual active site and keeping it getting in there the normal way, or non-competitive, binding somewhere else, changing the shape, and then you can't get in. So there's some pictures for it. Oh, what I just a pen. I'm just right, so competitive, literally your normal is this darker color one. The competitive inhibitor is a similar shape, but it's not the correct molecule, but it comes in and binds, and now the normal molecule wouldn't be able to bind there, right? If it wanted to. That's competitive. Non-competitive, we have our normal guy binding into its active site. But if this regulatory molecule on the regulatory site on the side comes in, it changes the shape of the molecule by doing that. Now our guy can't bind at its normal site. You see how it's changed its shape. Non-competitive, so it didn't actually bind at that site. Okay, so enzyme repression is just having some sort of feedback back to the cell to regulate further how your enzyme is either being made or whether or not you're making the enzyme. But you can imagine it from the genetic standpoint. We have enough of a molecule made, and then we can have, binding on the DNA and blocking transcription and translation, which we kind of talked about with the operon, which is why I'm not going to go into too much detail here. But there's a lot of levels that you can actually control your enzyme production as well as function, is what I'm trying to say. So not just the inhibitors, right? But controlling the gene expression as well. That's all it's saying. Okay. We get induction and repression a little bit. So different kinds of energy-based reactions in cells. We have exergonic versus endergonic. Exergonic, so just think X being like exo. We are going to be releasing energy. Ender being endo, requiring the addition of energy. Exergonic reactions are reactions that, like if you're doing them in chemistry, exergonic that release energy are going to be getting warm, right? They're releasing heat. Endergonic reactions, if you're doing them in chemistry, they're going to be sucking heat out of the environment, which means they're getting cold to your touch. That's the difference between these here, where they are releasing energy or using energy is basically all it's talking about. So coming back to our redox reaction. Going back to our redox reactions, everything is going to occur with pairs, right? So we always have something donating an electron, and the other thing will be accepting the electron, always, every time. So electron donor and electron acceptor. And then the oxidoreductases are the enzymes that are going to be helping facilitate that switch. And then we have our coenzyme carriers that are going to interact with those, because when the coenzyme interact with enzymes to help them function. They're cofactors that are organic. So for these guys, and I'm not going to make you know what they mean, and I'm not going to make you know the structure. So you're welcome for that. Are yeah, NAD and FAD. Again, I know I've mentioned them, but like I can't tell you how important they're about to get. So let's go over our redox reactions with our little picture here. We love talking about, our ionic bonds between our sodium and our chloride. But now let's talk about their actual donation of their electrons. We know how it's going to work. We talked about it by like looking into it just briefly with our ionic bonds. But if sodium has this extra electron in an extra orbit, right? This is kind of unstable. Most molecules don't want to have just one electron chilling in an orbit by itself, especially when that's an orbit that usually wants 8. So why would it want that? Whenever we have chlorine that has 7 here, so it's just dying for that 8 to help it complete. It makes sense now why they would want to pair up, yeah? So they don't just create a bond to one another like a covalent bond and share it. They don't need to, they don't want to, they're happier just getting rid of it. So that's why we have that ionic bond happening. When you have that though, now we have the trading of electrons. So sodium is going to lose its electron and chlorine is going to be gaining. Don't worry about reducing agents and oxidation. I'm never going to ask you about it. I want you to just really focus on oxidation or reduction, okay? So sodium is going to lose, which it obviously does here. Look, it lost it, right? Lose electron, oxidize. I say Leo, says Gur. You do what you want to do. And then reduced. And I am so a proponent of like remembering Leo says Gur or oil rig, that even when I'm looking at multiple chemical things and I have to know which one is oxidized or reduced, I will write it down. And it'll be like, okay, was this reduced thing, blah, blah, blah. And I'll be like, okay, what does reduced mean? Leo says Gur. Oh, all right, gaining electrons. So that's what, if that's a fine, man, write it down if you need to, because I still do. So oxidized, Leo says Gur, so losing electrons, we lost our electron here for sodium, right? We see where it went, it's gone now. So we have a positive charge there. And then chlorine gained 1, so it's happier out here with the 8 instead of the 7. So it gained, so Leo says Gur, gain electron reduced. So it's negatively charged. And that makes sense now that we've applied this just to that simple concept of looking at our ionic bonds, which we already knew was going on between sodium and chloride. That's all that's going on here. We're just like describing where the electrons are moving around in that bond. Okay. But we're going to talk about, they're having some good times over there, I guess. We're going to talk about moving electrons in a much bigger picture and throughout a lot of reactions. Let's just put it that way. Okay. So anytime that you are taking an electron from a molecule, that electron basically can serve as a source of energy. It stores energy within itself. It's an active unit and stuff like that. So most of the time, when we're looking at electron storage, we're going to store it in the form of hydrogens. Okay, so we know all about how electron is one like kind of unit. And if we were looking at just hydrogen by itself, we know it has one proton. We always talk about being a proton, but it has one proton and it has one electron. Only when it loses that electron is it H plus, right? Our proton, a proper proton, an H plus ion. So normally it has one electron and one proton. This is negative and this would be positive. So we can use that as a means to transport electrons instead of just rando free electrons, which we'll have those too. But a lot of times we're going to be going from, you'll see us go from NAD to NADH. That means we gained an electron because it came with the hydrogen. Okay, so that's what we're about to get into. You'll also see that when we do that, we often will have an H plus that will come along with that. That has to just do with the chemical reactions. They're important too. Those H pluses are hella important as well. We'll get into those too. All right, so hydrogens have a proton and an electron. We need those for our, be able to switch our electrons around in our molecules. Removal of hydrogens during a redox reaction is called dehydrogenation, go figure. And then I don't know what to say about it other than that. When we're going to use dehydrogenation as a means to supply electrons in our electron transport chain, which is our very last stop in the aerobic respiration, the creation of ATP. All right, so we already talked about NAD and FAD. There's also NADP. This is really only important in photosynthesis. Yeah, we're going to talk about that today. You better believe it. So really for now, starting off, I want you to more focus on NAD and FAD. Like I said, NAD is going to become NADH, usually with an H plus. Not always, depends on the chemical reaction. And then FAD is usually FADH or FADH2. Just depends on how you're looking at it. But like, I'm not ever going to quiz you on the differences between those, okay? What I want you guys to bring out of this discussion when I'm talking about these electron carriers, be able to recognize NAD or FAD as our electron carriers. And if there's an H on it somewhere, it has been reduced, right? Gain electron reduced. Okay, now we're getting it. Catabolic pathways, we're going to be breaking down things to steal off electrons essentially, so that we can use those later on to create energy. When we're talking about aerobic metabolism, when we're shuttling our electrons through everything, the final end product, we'll have some electrons chilling. We can't just let them just keep chilling, right? They have to go somewhere. We have to clean that mess up. And oxygen is going to do that for us in aerobic respiration. We have other organisms that don't use oxygen, and so they'll use a different compound. So go figure. Something else. Wow, they're not going to use oxygen. Guess what they're going to use? Not oxygen. So we have our ATP. Now we're really getting into this. We've talked about this being adenine, our nitrogenous base, ribose, our 5-carbon sugar, and our 3 phosphate groups bound to that ribose. Those phosphate groups are highly negatively charged. They are very bulky, and they are always repelling one another. So if we remove a phosphate, we release energy. And this is what's basically going to be powering the cell later on. So we need this to help power the cell. And it's so important that, you know, we're constantly making this in our bodies. Anyways, so moving on, it's definitely our primary energy currency in all cells that we ever have found that we know of. Okay, so it's always, if you're alive, you use this. And we can generate this in a couple of ways. This could be via substrate level phosphorylation, which just means moving a phosphate from some other piece of chemical onto ADP to turn it into ATP. So we can move that. So like a transferase, remember how we talked about that? Transferases can move the phosphates even. We can also have oxidative phosphorylation, which is pretty much the focus of what we're going to be doing today when we get into aerobic respiration. This just means we're using redox reactions to drive the production of ATP. We're going to talk how that works, of course. Then photophosphorylation, of course, is going to be making ATP that's driven by the energy of the sun. That's what plants do, not us. I want to do questions. No, I'm gonna wait till the end. We'll do the end one because I feel like that one's more important. Okay, if we have time. Of course, we're probably not going to, right? So you can just like not worry about it. All right. So catabolism, breaking down things. Right, cats destroy. Catabolism, our catabolic pathways that we're going to be learning about now. Aerobic respiration, anaerobic respiration, fermentation, and yes, you do need to know about all of them. When we get into the lab, we're actually going to be looking at these processes, and you won't even realize that we're looking at these processes. Now, I'm going to be relying on these a lot, especially the fermentation, because a lot of bacteria go through fermentation. That can't be a surprise. So the number one starting process for all of this is called glycolysis. All of these guys are going to always start with glycolysis. Now, this is aerobic respiration pathway, anaerobic, which is anaerobic, should tell you without air, no oxygen. Air, that's oxygen. And then fermentation, which is its own little separate kind of guy on the side, right? So all of them start with glycolysis. We're going to go over what that is and what it means and how it works for these cells in a minute. But notice that fermentation don't got a lot going on after that, right? So basically just goes through that, and then its products that it makes from that, it has to deal with those, and then that makes things like acid, like lactic acid, if you're making kimchi or kombucha, or making alcohol, carbon dioxide that's in your beer, things like that will come from fermentation, which is how they deal with their byproducts of glycolysis. Okay. Then if we compare aerobic versus anaerobic, right? We have the next step, the Krebs cycle. They both have that. Wow, that's not different at all. The only thing that differs is down in the electron transport system, electron transport chain. I call it the electron transport chain, ETC. So down here in the electron transport chain, aerobic guys use oxygen. Anaerobic guys do not. So they use whatever. They use these guys, for example, not oxygen. That's the only difference. But now I want you guys to take a look at, if I give you one glucose molecule, just starting with like one glucose molecule, right? All of them are. One glucose molecule, whatever, glucose, in aerobic respiration can yield 36 to 38 ATP. The bacteria get 38, we get 36. We lose some energy because we have to transport things across the mitochondria. They don't have a mitochondria, so lucky them. They are our mitochondria. So we have 2 to 36, depending on what type of anaerobic respiration you're dealing with. Still could be not too bad, but look at fermentation, y'all. They only get 2 ATP for every single glucose molecule they come into contact with. That's like nothing. So now we wonder why they have to go through so much glucose and create so much alcohol or acid or carbon dioxide for us to get their ATP, their energy, just to survive. So it's pretty useful for us, but not particularly for them. So fermentation is only going to be done by facultative and aerotolerant anaerobes. I'm never going to ask you about the aerotolerant guys in this purpose. I'm only ever going to ask you about facultative for fermentation. Facultative fermentation, F and F, right? Facultative. We talk about obligate this and obligate that. Obligate meaning you have to do these things. Facultative anaerobes can also be called facultative aerobes. It doesn't matter. All it means is they can switch between using oxygen, and when oxygen is not present, they can ferment. So it's their option when there's no oxygen. So oxygen isn't required, and they can use organic compounds basically in their glycolysis process as their electron acceptors to just help them make those two little measly ATP and then cycle through. So pretty lame as far as like the output of ATP, but it's a very good trick that they have if they don't have oxygen available that they can. If anybody knows anything about brewing at all, then you know that you have to balance basically your exposure of like your yeast or your bacteria, which yeast ferment, it's the same process. It's just a different organism. So yeast or bacteria, you have to balance the oxygen within their environment versus, not oxygen. So you can push them to multiply, which they're going to do more of in the presence of oxygen, right? There's more energy available. But once they multiply and you want them to make your products, you want to get all that oxygen out of there and force them to ferment. So you can control that process in brewing. And they do, obviously. So if you do not have oxygen available and you are facultative, you can go through fermentation to make your energy when there's no oxygen. But of course, we already saw having oxygen available, going through that aerobic pathway is the best way to make energy. It gives the most by far. All right, so aerobic respiration is just a whole bunch of catalyzed reactions that are going to line up together to allow us to make ATP. Our first step in that process is glycolysis. If it sounds like it's talking about glyco being sugar and lysis being like we lyse a cell, breaking apart, right? Busting open, like that sort of stuff. We are in fact doing that to glucose. Glycolysis is the breakdown of glucose, and that is our main compound that's going to be going into this. Glucose will be converted to pyruvic acid. Glucose converted to pyruvic acid. That's the main what's going on. All right. Now let me introduce you to what is going on in the middle of all that, which is you're not going to like me for it. This is always the first phase of any of those energy producing pathways, aerobic, anaerobic, or fermentation. Okay. This is what it looks like. This is glycolysis. So when I'm telling y'all that it's a lot, like I'm like, oh yeah, okay, glucose to pyruvic acid, whatever, sure, show me. There you go. So I'm not going to make you know the steps. Again, you're welcome. I think a lot of micro classes will require you to do this, but I don't think that that's important. If you are interested in the pathways of this is something you learn in biochem. So if you're going on further from here, you're going on to your BSN or something, it might be nice to take that as a supplementary course if you're interested in metabolism. But if you're not, this is a lot, right? Basically, all that's going on is we're taking glucose, which is a 6 carbon. You can see it here. I like that they did at least do that. 1, 2, 3, 4, 5, 6. 6 carbon compound. All right. And then we're taking it through a series of steps where we're going to be moving around some molecules, moving around some electrons and all of this stuff, all the way down to, you can see already here, we've got 3 carbon and 3 carbon, right? And as we're switching it up, eventually, y'all, we get down to pyruvic acid. Okay. So we've gone from 1 6 carbon glucose to 2 3 carbon pyruvate molecules. We have not lost a single carbon, but in the process, we've moved around our electron situation, made it more a little bit efficient for the cell. And we have created a little bit of ATP here. Overall ends up being like 2 ATP total, because even though there's 4 here, because we go on each side, and then we used 2 up here though, just so you see. Okay. So overall, a net of +2 ATP here. That's what's going on here. And I want you to notice one other thing as we're doing this BS. Okay, this thing here, what have we done here? Now we call that NAD has been reduced. And that's important because we're going to come back to these guys. Okay, see how it says over here on the side, 2 electron transport. That's the goals, man. So anyways, I always start off, we look at this and we can see that there is a big picture thing going on here. We're shuffling around electrons and bonds and all this. But for your purposes, what is glycolysis doing? It is going from our 6 carbon glucose, our starting molecule, 6 carbon glucose, to 2 3 carbon pyruvic acids. Okay? That's the big picture thing. If you can remember that there's ATP made here, I mean, I'd recommend remembering that there is some ATP, but very little, because of course it does, because that's all that fermentation gets their ATP from. It has some, not very much though. All right, so our pyruvic acid, we call it a central metabolite because it's not only going to be important for our next steps in making ATP, but we can use it for a lot of other steps in building molecules for the cell. Pyruvic acid is very important for this. If you're fermenting things, then that's where you would stop. You would have your pyruvic acid as your end product. But you remember how we made those NADHs? We reduced the NAD to NADH, right? Well, they don't go, if you're a fermenter and you made NADH, that doesn't go to the electron transport chain. I'll tell you why. Because the electron transport chain relies on oxygen to do its work. If you don't have oxygen, you cannot go to that lovely land of electron transport chain. So what do they do with their electrons? Save it for a better day? Well, that's not really how it works. So what they do is they shuffle around bonds and jump like that. And that's where you get your alcohol and your carbon dioxide and everything else, okay? So we don't just have pyruvic acid. We have to we'll shove our electrons back into that and make these byproducts. Depending on what organism you are, depends on what byproduct you're gonna make, okay? That's basically what's going on there. All right, so strict aerobes like ourselves, we call them obligate aerobes, and some of our anaerobes are gonna go through Krebs next. But first, we're going to go shift it into a different kind of molecule. What do I want to say? This is just summarizing what's going to go on in Krebs. Because again, this is not going to be another user-friendly thing, okay? Krebs, called the Krebs cycle. It's also called the citric acid cycle. And we're going to take our pyruvate and cycle it through. And the biggest picture going on with the Krebs cycle is we're going to be making electron carriers like NADH. Okay, so we're going to be reducing a lot of things. That's the goals, all right? So each one is going to go, each pyruvate, because we made 2 from our glucose, each one is going to go through the Krebs cycle. And we can make 2 ATP from that during the process. Again, a little minor amount of ATP that nobody really cares about, because that's not the goal. We want big numbers. So here's what we do, y'all. This is your pyruvic acid. We're going to turn it into this molecule called acetyl-CoA. I want you to notice something about acetyl-CoA here. We have 3 carbons, 1, 2, 3 for pyruvate. How many carbons does acetyl-CoA have? 2. Where does that carbon go?
Oh.
Carbon dioxide. It's released as carbon dioxide, all right? Then, all right, so we'll come back to that in a little bit, I promise. But then we'll go in through the Krebs cycle, blah, 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 all this way, okay? Don't need to know the steps of it. Once again, this is why it's called the citric acid cycle, just FYI, in case you ever hear it called that again. But that's why it's our first molecule in the Krebs cycle. But while we're shifting around all these freaking molecules and recombining, because you see here, we've got this 4 carbon over here, that's right here. It's going to come bind up with our 2 carbon here. And then we're going to make that a 6 and shuffle everything around, literally just moving bonds around. And as we're doing that, stealing off whatever electrons we can. And occasionally we'll kick off a carbon in the form of carbon dioxide, right? So we kicked off a carbon dioxide up here at the top by acetyl-CoA, and we're going to kick one off here. So that became a 5 carbon now, and then we kick one off here. So then now we're in the 4 carbon territory. All right, so we did not make any carbon in glycolysis, right? We didn't make carbon dioxide in glycolysis because it went from 6 carbons to 2, 3 carbons. We didn't lose any. Here in this process, in the Krebs cycle, it's important to know that because that might be something that's on the test. Do you make carbon dioxide in glycolysis? Do you make carbon dioxide in Krebs? You should know those major products. I might not ask you the numbers exactly, but be aware of what you did make or didn't make. Okay. So here we're making a bunch of these reduced electron carriers. This is the goal of the Krebs cycle. So we're moving around all these molecules so that we can steal off these guys and this guy here. There's another one over here, FADH2, like I told you was coming, right? This guy here. We've made this guy up here. Look at how many we made from just 1 pyruvic acid molecule. And y'all, we got 2 from 1 glucose. This is just one of them going through. So we'll go through again with the other, right? So we're getting a whole bunch of these NADHs and FADH2s to carry our electrons into the next step, which is the big daddy step, all right? What we need to know about glycolysis, you went from glucose to pyruvic acid, made a little bit of ATP and a little bit electron carrier. Then we turned our pyruvic acid, that we got 2 of, from that into acetyl-CoA, lost a carbon dioxide, and shuttled what was left through. Lost some more carbon dioxides, made some NADHs in the process. Okay, FADH2 as well. That's the picture. That really is what I want you to know about this. I don't need you to know the intermediates. Okay, so now we're going to go through the electron transport chain, also known as the respiratory chain, also known as the electron transport system. This is where we're going to shuttle our electrons through and make our ATP, right? This is our ATP factory. All right, so this is what it looks like on a bacterial membrane. So on their actual cytoplasmic membrane, which remember that's their inner membrane, they have all these little protein guys here. And our NADH is going to pop off an electron and it's going to leave behind an H+, okay? The electrons are going to move through these guys, these proteins, and as they're exciting these proteins, these H+ get shuttled out. You see how many of them are going out? So you get all these H+, 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 plus on the outside. It's not in the wall, it's on the outside, okay, just drawing it there for this purpose. This happens in our mitochondria as well, but it's going to happen on those folds, those cristae, okay? So we're doing this. We're shuttling these negatively charged electrons through so we can pump hydrogens out. This is a process. This means of moving H+ out because of electrons exciting, I don't know, these proteins, and they're called cytochromes. So I'll just write that so that whatever you've heard it. So as those electrons are negatively charged moving through those guys, it causes those positive guys to pop through the other side, right, as that's going. This is called chemiosmosis. Okay. This is incredibly important for what's about to happen next, all right? All right, so you create this gradient of pluses outside of the cell. And we think of it, we talked about this already with diffusion and gradients and all of this. So if you have a dam, think of it as a dam that is holding back all of these H+, yeah? And it's plus charge. And so all these plus charges want to equal out on both sides. They don't want to be plus on this side and not on the other side. They want it to be equal, just like everything always does because physics, right? They want to equal out, but there's only one way to go. So they're being held back by this dam. And the only way that they can get back into the cell to equal out is through ATP synthase. ATP synthase, in case you can't tell by the name, is an enzyme that's going to create ATP. When those positively charged hydrogens, we've talked about proteins, y'all, and how important those charges, those pHs and everything can be to the structure of those proteins. You get pluses driving through these proteins, and that is going to cause change in that protein that allows us to phosphorylate ADP into ATP. And that's how we drive ATP production without having to put in energy to do it. We've just used a gradient. That's all we did. And so, anyway, so it's cool stuff, but that ATP synthase will drive that ATP production. And so you always have to have that H+ on the outside to have that going on, right? So nice. But when you move H+ back into the cell, they're going to be back in the cell. So what happens if you start building up those H+ back inside the cell? You can't have them still chilling around, right? You have to keep that gradient outside. You have to keep that dam with all that pressure to drive that motor to make your ATP. So what we do is you've made your ATP. We're collecting our H+. You breathe in oxygen. And those H+ with these electrons that came through this whole process, H+, and we'll have an H+, we'll have an electron, and we'll have an electron, and we'll have half an O2. And then that all together will make H2O, right? That's how oxygen handles the gradient problem. Oxygen comes in and it takes care of those buildup of those H+, but it also takes care of the electrons. It's the final electron acceptor. So if I ever ask you guys, what's the final electron acceptor in aerobic respiration? It's oxygen. First of all, you should automatically think it because I'm saying arrow, that should give it away, right? Arrow, oxygen. So we breathe in air, we breathe in oxygen. If we don't have oxygen in the air, it doesn't help us, right? We have to have the oxygen. That's what we need. This here that I just explained to you, all of these steps, going from glucose into the pyruvic acid, going from pyruvic acid into basically all these electron carriers, giving off carbon dioxide in the meantime, putting those electrons through here, pumping the electrons outside to create the gradient, and then bringing them back in, creating ATP, but eliminating them by splitting water and splitting oxygen, making water, right, as our product. All of that. That is why you breathe in air, oxygen. That's why you have to breathe in. Because if you don't breathe in, that gradient will disappear. We'll just have H's on both sides. And if you don't exhale carbon dioxide, then you have buildup of that. Of course, I mean, that's one thing, but like the whole reason you breathe out carbon dioxide is here. So now when I tell you guys that the carbon dioxide, everybody always thinks they breathe in oxygen, and the oxygen molecules that are in the oxygen that they breathed in are the ones that are in the carbon dioxide that they breathe out. And it is not. You've seen that now. The carbon dioxide that you breathe out is from your food. It's from your glucose. And then the air that you're breathing in immediately goes to making water, which is why even if you are like in the desert and you haven't had anything to drink, if you have to be alive and have energy to do life, you will always have urine output because you have water output. Always. This is why also, not just this, but like the electron transport chain, when I say to you guys, and I've said it before and I say it jokingly, I do mean it jokingly, just FYI. If I suffocate you with a pillow right here and you die in a matter of a couple minutes, that's why. Because you're not getting oxygen. Isn't that something, y'all? People don't think about it being like that, but now you've learned a thing today. Congratulations. This is why I like this chapter, because I feel like it just brings everything so full circle in one go. I hope that you guys think this is just as cool as I do. Here's a picture of how it would work on your mitochondria. Those molecules would be lined up on your actual folds of your cristae. Okay, so we made ATP via a process that we call oxidative phosphorylation. We mentioned it earlier, but this oxidative phosphorylation, this is oxidative because we're moving electrons through that electron transport chain. Okay, so that's why. So that's how we're making our ATP. We called that, I told you what chemiosmosis was, and the proton, that force, that buildup of the hydrogen ions on the back of the dam that we always have to keep higher on the inside, right? That's called the proton motive force. So think of it as protons are giving the whole motivation as a force for making ATP. If they are not there, then you're not going to have ATP production. So proton motive force, that charge on the outside being positive and inside being, relatively speaking, negative, that's what's going to store potential energy. And if you guys don't know what potential energy and why that is, it is exactly like a dam. Or if you have literal actual water molecules behind a dam and it is like wanting to, all the time, it wants to burst, right? If you break that dam, what's it going to do? Everywhere, right? All that energy that you held back. Or you can use that energy to power motors. That's what is going on here for us. Potential energy is that. And potential energy is also like when you're holding up a pencil, like I think I've explained it before, like if you're holding up a pencil, you drop it, I just released the potential energy that was held in that pen while I was sitting up here, wanting to follow the whole time. That's all it is. We'll use that to drive ATP production, not just once, but constantly, all the time, in all of your cells, always. All right. We already talked about this with the mitochondria. These are some summarized versions of what we were talking about here. I just want to get into the photosynthesis, but if you guys want to look at the overall production and products of these things and what is produced in what state and whatever, or what, yes, what is produced for what step, these are some good ones to look at, but we already talked about this one. In anaerobic, like I said, it's just not going to be using oxygen. It'll use something else. We don't ever go through any of that with fermentation. Fermentation only goes from pyruvic acid. And then when it stops there, this like NADH problem that we have, it has to get rid of it somehow. So it can either do it by making lactic acid or alcohol or acetaldehyde or something. Depends on the organism. Okay, so you can also break down fatty acids. So the reason your body stores your energy as fat is because oxidation of a 6-carbon fatty acid that's stored in your fat cells right now, 6-carbon fatty acid versus a 6-carbon sugar, glucose, that is what we just looked at. 50 ATP from the fatty acid, as opposed to 38 for glucose. That's why. So your body's going to store it that way because it gets more energy from that 6-carbon molecule later on when it uses it, which is not great for you because then it's like has all this energy stored that way, and then you're not really using, giving more energy from that one state than you would from the other thing. And the whole purpose of like going into keto is basically like to have your body burning energy while you're also making energy, like not storing too much of this, getting the amount of amount of energy that you need from having not too much calories and excess energy. But it's opposite with the fats. So anyways, you can also break down proteins through using proteases, enzymes that break down proteins. And these intermediates can go into any of those steps as well. We can also take any of our products from any of those steps and use them to make other molecules. This whole concept of entering in with other molecules or leaving any step with molecules to build other things is called amphibolism. So amphibolism, going both ways, you'll be breaking down and making things all the time. Of course, whatever the body's going to need from whatever step it can take, however it needs it. But that's amphibolism for cell efficiency. Yeah, of course, you're going to be needing to build things if you're making new cells and whatever. Cool. I'm sure you guys can put 2 and 2 together with that. I just wanted to get to this before. So we have photosynthesis. We have 2 reactions that photosynthesis will go through. Light dependent and light independent. Light dependent, of course, is only going to be present if sunlight is present. Okay. Light independent, it doesn't matter if there's light or not. It could be in the dark. They used to call them the dark reactions, but now we just call them the light independent reactions. All right. So these light dependent guys are catabolic. So they're going to produce energy. The light independent guys, they don't need light. And they're going to be anabolic. So we are synthesizing. So let's talk about what's going on here. All right, so light dependent reactions. Was there a picture? Not really. Okay, great. So light dependent reactions, it's similar to the chemiosmosis. You are going to have molecules next to one another and whatever, and we're going to excite something and cause electrons to move through, create H's that come out, right? Same thing. We just saw this, right? It seems familiar, except our excited electrons don't come from NADH or whatever. This comes from the sun. The sun excites molecules like chlorophyll, excites electrons from that, from the chlorophyll, and those get shuttled through, okay? So instead of NADH or whatever, the sun excites chlorophyll, and that creates excited electrons that move through, and then eventually you're going to be making, through ATP synthase, it's the same process otherwise, ATP. It's not very much though, just a little bit. But you know what it is enough for? It's enough to drive the dark reactions. In the dark reactions, what we're going to be doing is taking carbon dioxide and essentially through these processes that you don't really need to worry about, from carbon dioxide, we're going to make a 6-carbon glucose using this ATP. So you make 2 ATP with your sun. I guess there's the third one here. You make a little bit of ATP with your sunlight, and you're going to crank it into the dark cycles or the light independent cycles. They're also called the Calvin cycles. Do you like how many words there are, like names for these things? The light independent ones, though, we already made our ATP from the light. Now that we have it, we're going to use that ATP to fix carbon into glucose. That's what we're doing. That's what plants are going to do. So why would plants need to make glucose? Well, first of all, they don't get any organic molecules from their food. They make them all. So that's their setup for that. So they need a lot of glucose just to make their organic molecules. The other thing that they need it for, because we're going to be making all this stuff, we just used up all our ATP from the sun, by the way. Do that. So in order to make our new cells and make our new molecules with our glucose and do all this stuff with our cells, what are we gonna need? We're gonna need ATP, right? You wanna know how they get ATP, y'all? Same damn way you do. They use oxygen to do it. People don't know that about plants, but yes, they use oxygen too. They don't just make it. They make an excess comparatively speaking. You know what I'm saying? So they still go through aerobic respiration, y'all. Is that not cool? Also, they make their own glucose to pump into it though. They don't eat theirs. I think that's just the coolest of all time. So that's it. We'll go to lab now. Thanks for sitting through my coolest chapter. None of the other ones are going to be this cool. Sorry.
Right, let's just watch like the Tuesday lecture.
Hopefully I'll have them on Tuesday. If I don't and you guys want them, just let me know and I'll give them to you. Yeah.
I'm letting you know.
Yeah, just email me for it. I don't mind that because I leave them out ahead of time so that you guys don't like do them ahead of time. Yeah. But yeah, afterwards I don't mind if you guys want to have that. All right, I'm gonna stop this. I'm gonna go to the lab.